The Impact of Lyme Disease by Carlene Keene. Lyme disease is the most commonly reported vector-borne pathogen that is carried by black-legged ticks. These ticks go through 3 stages of life over a 2-year period: larva, nymph, and adult tick. The nymph stage is the greatest threat to humans because they are able to transmit the disease without a trace. Some hosts of these ticks include the white-footed mouse and species of deer. Lyme disease is reported by about 30,000 people each year in the U.S. However, there are many cases that go undiagnosed. The real estimate may be around 300,000 infected people. This disease may cause a rash around the bite, migraines, pain or swelling of large joints, etc. This is a bacterial disease that infects large populations of the U.S. and around the world. There are some factors that may reduce the risk and spread of Lyme disease. Primary prevention may be to stay out of woody and tall grass areas as they carry many ticks. Some environmental factors that will help are biodiversity, intact forests, and the control of acorns. These determinants may be able to save a lot of lives if ways of prevention are known for more people and organizations. My research consisted of 4 main questions and topics. What are the patterns and locations of Lyme disease from 2000 to 2015 in the U.S., and how does it spread over time? Create a county-level database with the land features using zonal statistics. Find correlations using scatter plots to show similarities between Lyme disease and the landscape features. And using grouping analysis, what patterns returned and what does the future of Lyme disease look like? This is a motion picture of the density of Lyme disease from 2000 to 2015. You can see the major areas that are red buffer in the same 2 areas, the upper Midwest and the East Coast. There are some outliers around the U.S. that can be explained by tourists visiting these high-density sites and bringing it back to the West. As time goes on, Lyme disease densities expand and retract around the U.S., especially towards the south. The high-level densities can be explained by climate change and lack of primary prevention to infected humans. This model represents the GIS of how each layer was made. I started by reclassifying the 3 input features which change the values in the raster. I used the zonal statistics tool for my 3 variables, slope, deciduous trees, and evergreen trees, to calculate statistics on these values of a raster within the zones of another dataset. The end result of this model was to use zonal statistics to predict if there is a pattern between Lyme disease and landscape features we know. This map displays slope of the U.S. The red area is where there is the highest slope, which could be interpreted as a mountain range. The yellow is then flat plains. This scatter plot shows that at lower slope values, there are higher densities of Lyme disease. This can be explained by urbanization in these flat plains, such as the Midwest, where a large population of people live with the disease. Also, in the middle slope range between 1 to 2.5, there are many people on the East Coast that also have the disease. The highest slopes have 0 cases of Lyme disease because most people and diseases don't thrive on the mountaintops. These high areas would be wild and controlled of diseases. Overall, when slope is moderately low, Lyme disease is high in certain areas of the U.S. This map depicts the humidity range from high humidity in blue down to red as low humidity. The high humidity area contains habitats for trees, deer, mice, and ticks. These species are thriving in the moist areas where Lyme disease is prevalent. The low humidity areas have desert-like air, so the ticks would dry out and not live to spread the disease. The humidity scatter plot reiterates the fact that the high humidity areas in blue and green have the highest values of Lyme disease. The red or low humidity areas have 0 cases of Lyme disease because the ticks can't thrive there. The deciduous tree map details the high density in dark green while low covers of deciduous trees are in light green. The areas of dark green or high amounts of deciduous correlate with the areas of high Lyme disease densities. The deciduous scatterplot can tell us a lot about the range of Lyme disease. The low and high values of deciduous trees have 0 cases of Lyme disease because there are either no trees or there are very healthy forests with boxes to eat the mice that carry the disease. The middle area has the highest amounts of Lyme disease because there are no foxes, a lot of mice, high urbanization, and fragmented deciduous forests. These are areas where ticks thrive and can easily transmit the disease to people with little to no predators. This map shows grouping analysis of the landscape features, which groups features based on feature attributes and optional spatial or temporal constraints. The features I used were elevation, slope, tree canopy, fragmentation, deciduous, and evergreen trees with no spatial constraints. The grouping analysis linked the traits into 3 main categories: blue, red, and green. The red areas can depict low levels of Lyme disease. The blue areas can depict high levels of Lyme disease. I didn't add any previous data about Lyme disease, yet the correlation that this map has to the density map is parallel. The relationship here is strong, which means the grouping analysis returned a pattern even with no Lyme disease data. I can use this grouping analysis to predict future patterns of Lyme disease that may spread to the south or other blue areas. There may be future research or questions available here to know whether climate change, biodiversity loss, or other factors will excel or stop the spread of Lyme disease before it is too late. Thank you for watching.